Hi, also, I forgot one last slide about caffeine in the last video. So ultimately, why caffeine is said to ultimately help increase lipolysis or hormone-sensitive lipase. Ultimately, it's thought to antagonize adenosine receptors. So it acts as a competitive antagonist for adenosine. And what does adenosine do? It ultimately reduces the sensitivity of adipocytes to norepinephrine and ultimately reduces lipolysis. So if we have a competition between adenosine and caffeine wins over, the thought is that, okay, we can actually increase lipolysis and norepinephrine can do its job. However, if adenosine were to win out, adenosine attaches to those receptors and norepinephrine is not allowed to do its job, and it reduces the active lipolysis for the most part. Okay, so now we're going to move on to the markers for lipolysis. So as a result of lipolysis, glycerol levels will rise, right? Because we talk about the tag, we have a glycerol backbone and then 3 fatty acids. So if we're trying to access those fatty acids to use them to break them down for energy for ATP, we're going to have a lot of glycerol left over from that triglyceride. So ultimately, a good marker for lipolysis would be high levels of glycerol floating through the bloodstream. So that means we're burning a lot of fat and we're accessing a lot of triglycerides. So ultimately, we'll have a lot of free glycerol floating around. Glycerol can be shuttled to the liver to participate in gluconeogenesis or glycolysis. Ultimately, it does this by being converted to G3P and DHAP and to gluconeogenesis or glycolysis. So we can use it actually in other metabolic pathways. The rate of glycerol released by fat cells is greater than the rate at which the liver can use it. So that's why we're going to have, if we took a blood sample, we would see high levels of glycerol in the bloodstream. Because even though we're shuttling a lot over to the liver to be used in other pathways such as gluconeogenesis or glycolysis, we're breaking it down too quickly for our liver to use it at that same rate. So that's why a good marker, if I took a blood sample, and if I were measuring glycerol levels, I could indicate if you're going through high amounts of lipolysis or not. Okay. It will rise also faster than the rate of free fatty acids, and plasma levels of glycerol will again give an indication of lipolysis, but also free fatty acid levels, an increase in those as well, will show an increase in lipolysis and us accessing our triglycerides. So glycerol is thought to be a good rough indicator because it's not used by muscle to any appreciable extent, since muscle ultimately lacks glycerol kinase. It's also true that the adipocytes lack the enzyme, like I just said, glycerol kinase, and therefore cannot use glycerol as a substrate within the muscle tissue itself. What does glycerol kinase do? It's involved in triglyceride and I'm going to spell it for you, GLY-CERO phospholipid synthesis. So it catalyzes the transfer of a phosphate to a glycerol, forming glycerol 3-phosphate. And ATP plus glycerol, ultimately, when we're looking down here, we're going to look now at this metabolic equation. So ATP plus glycerol using glycerol kinase can produce an ADP and glycerol 3-phosphate. This substrate becomes a significant source of glucose in hibernating animals. So here's a fun fact for you, aka the black bear, where lipolysis becomes necessary for survival. So ultimately, this is a nice little hibernation metabolic equation for you and how hibernation kind of works. Okay, so mobilization. The amount mobilized. This is our last bit about this step. The amount of fat mobilized depends on a number of different things. One, the rate of lipolysis, how fast are we breaking down fat. Free fatty acid carrying capacity of the blood as it flows past the adipocyte. Reesterification and lactate, since lactate inhibits lipolysis, right? So ultimately lactate inhibits lipolysis like we talked about a little bit before. All right, so now we're in step 2 of this whole 7-step process of lipolysis. We're going to talk about circulation. Ultimately, now we've accessed our triglycerides, we've accessed those free fatty acids, we've broken them down using desnutrient, hormone-sensitive lipase, and now we've broken them apart from their glycerol backbone. So how do they travel in the blood? That's ultimately what circulation is referring to. How are they traveling? Most, which is 99.9% of the free fatty acids in circulation, is bound to albumin. There's about as many as 8 to 10 binding sites on albumin for free fatty acids. However, only a few are occupied because the affinity is low. So even though there's 8 to 10 binding sites, they're not going to be completely occupied. All 8 to 10 binding sites won't be occupied. The affinity is technically low, so a few will be bound at any given point in time. But 99% of our free fatty acids in circulation will be bound to albumin. When there's an increase in free fatty acid levels, the binding sites are usually filled and the remainder is left as free in the circulation for the most part. Okay. The unbound portion of free fatty acids is in equilibrium with the bound form, ultimately. So what cannot be bound to albumin floating around the bloodstream is usually in equilibrium with the amount that is bound. So we do have some free floating in the bloodstream as well. During exercise, there is usually an initial small decrease in plasma free fatty acid concentration as the result of increased utilization. This is followed by a progressive increase as lipolysis and free fatty acids release increases and they accelerate. So I will say that again. So ultimately it makes sense. I'm at rest and then I start to exercise. And if I'm exercising up to my 60% VO2 max, not going more intense than that, so I'm at a low to moderate intensity exercise, you will see an initial small decrease in plasma free fatty acids. That makes sense because we're utilizing those. So our cells and so forth are saying, okay, we need to access those free floating fatty acids in our bloodstream. So we will see that initial decrease because that means we're going to need to utilize them in order for us to keep exercising. But afterwards, as we keep exercising, we need to produce more energy and access our adipocyte stores. So that's why it will be followed by a progressive increase as lipolysis is increasing during exercise. So then after you see that initial decrease of free fatty acid concentration floating in the blood, we will see a progressive increase due to the amount of fat tissue and adipose tissue we are breaking down to utilize for energy. So lastly about circulation, there's an increase circulating, our increase in circulating levels is mediated by a rise in the catecholamine levels. Again, our catecholamines are norepinephrine and epinephrine. Those are released by our adrenal medulla, which sit on top of our kidneys. And then also a fall of insulin. Okay, so step 3 in lipolysis is the uptake of fatty acids. So how are our cells going to uptake this in order to use it to break down to ATP to make energy? So the amount of free fatty acid uptake and oxidation is dependent upon the plasma levels of them. So during exercise, there's usually an initial, again, small decrease in plasma free fatty acids following by a progressive increase. The amount of free fatty acid uptake and oxidation is dependent upon these plasma levels. So that initial decrease and then the progressive increase. The uptake of those fatty acids, of course, obviously depend on those plasma levels. So that decrease and increase have a big impact on how much we're going to be uptaking within our cells. During exercise, the blood flow through the muscles increases because of dilation in the exercising muscles. So the greater the blood flow, the greater the delivery of free fatty acids will be to the muscle cells. So I'll say that again. During exercise, we're going to go through a little bit of hemodynamics later on in the semester, but blood flow is going to increase to those muscles due to passive vasodilation to those exercising muscles. So that increase in blood flow is going to cause a greater delivery of free fatty acids to the muscle cells. Makes sense? Increased blood flow. If we have an increase in free fatty acid concentration, the delivery of free fatty acids will increase as well as we exercise. Okay, so you can see here when we're talking about the uptake of fatty acids, the fatty acid again is bound to albumin mainly as we circulate through the bloodstream. But the fatty acid has to pass through the following layers, which is up here in this diagram for you. So we're in the bloodstream here. We got the fatty acid is attached to albumin. Fatty acid has to go through this microvascular space, number 1. We got to go through the endothelium, number 2, into an interstitial space. And then ultimately we have to cross the sarcolemma and then finally into the intramyocytal space, which ultimately is intramuscular. So within the muscle cells here, it has to get through. So ultimately this from the blood needs to get through 5 layers in order for our muscle cell to use this fatty acid to produce ATP, obviously then again to use free energy to perform mechanical work. So the uptake of fatty acids are as follows. Fatty acids are hydrophobic and therefore must be bound to a protein in order to be transported in the blood. So that makes sense. It's transported to albumin, so that's how it's able to circulate. Most tissues, including skeletal muscles, contain something called fatty acid binding protein. So you may see an abbreviation. It's called FABPS, and those are considered fatty acid binding proteins. So free fatty acid uptake consists of 2 components. One, simple diffusion, and saturable function of unbound free fatty acids. So ultimately, some transporters have been found in skeletal muscles and include plasma membrane fatty acid binding proteins, fatty acid translocase, which is so right here we're looking at this fatty acid translocase FATCD36. This has been known to kind of act like GLUT4. So remember we talked about GLUT4 last lecture. We talked about how during exercise GLUT4 is activated within the skeletal muscle cell and goes up to the sarcolemma and allows glucose into the cell and needs insulin there, allows to doing it as well. So the function of these transporters, for example, for FATP and FAT/CD36 is they are intracellular carriers to transport these fatty acids to the outer surface of the mitochondrial membrane. So ultimately they're transporters. This is how we can get free fatty acids into our muscle cell. We need these transporters in order for us to do that. Okay, so step 4 with lipolysis is called activation. So when fatty acids enter our cell, they're attached to a CoA, forming a CoA called fatty acyl-CoA. It is different than acetyl-CoA, right, because there's no T in here. This is called a fatty acyl-CoA. This reaction primarily occurs on the outer mitochondrial membrane. This is catalyzed by acyl-CoA synthase. Ultimately, this step requires 2 ATP molecules to activate 1 molecule of fatty acid. So that's important to know. Just like in glycolysis, we said there's an investment phase and a payment phase. Well, different portions of lipolysis require energy as well. So this is one of them. During activation, in order for us to activate 1 free fatty acid molecule, just 1, it requires 2 ATP molecules to get that activated. Okay, so here's ultimately where a diagram of activation of a free fatty acid. So we have the plasma membrane, right? Or we're in the plasma, so we're in the blood right here. And ultimately, we want this free fatty acid to come in to the muscle cells so we can also use it for the generation of ATP so we can keep exercising. So I don't need you to memorize this diagram. Ultimately, it's just showing you how many processes have to happen to just get 1 molecule of fat within our muscle cell so we can actually use it to generate ATP. So here we have acyl-CoA synthase, was what we were talking about. This is what helps activate this free fatty acid, and it requires ATP. Right, and then we're going to get into translocation next and on the next couple slides, and then we use a transporter protein, and we ultimately need to get back all this free fatty acid down into here in this mitochondrial matrix so we can actually use it. Okay, so on our next video, we're going to finish up the steps of lipolysis, which will be translocation, beta oxidation, as well as then finally step 7, oxidative phosphorylation. And then we're going to talk about a little bit about how endurance training affects lipolysis. And then lastly, we're going to finish up with a slide or 2 on protein metabolism and ultimately what I want you to get from these lectures. So click on the next video and we'll continue lipolysis.